Hello, this is Dr. Clark, and we're going to be talking about neurology today and the nervous system. So, the most basic part of the nervous system is going to be neurons, and these are the basic functional units throughout the nervous system. They have a very high demand for oxygen and glucose, and they're not actually able to do anaerobic metabolism. They have to have oxygen and they have to have glucose to be able to work. Past a certain age, they're not actually able to reproduce themselves. So if they get damaged or injured in any way in an adult animal or an animal past the growing period, they're not actually able to reproduce themselves. They're able to actually repair some damage further down on the neuron, but they're not actually able to totally repair the neuron if they're totally damaged. And a lot of your neurons are actually going to have support cells called neuroglial cells. These cells actually sit there and help to support the neuron in its function. So when you look at the neuron structure, you have dendrites up here. These actually receive the information in. Right here in the center, you have the nucleus and what we call the soma or the cell body. And then coming down is the axon. And so the axon is actually where you're going to have a lot of the myelination at, a lot of those neuroglial cells. And then at the end of the axons, they're actually going to branch off into what we call telodendrites. And at the end of each one of these telodendrites is a synaptic knob. Information comes into the nerve through the dendrites, so they're going to hook up to another nerve, or say something like a Panacea corpuscles, multiple ones at the skin. They're going to feel touch or pressure or heat or something, and they're going to send that information to the soma. The soma is going to determine if that needs to be passed on the axon. So you have these multiple dendrites. There can be 30, 40 dendrites on one single nerve. And then there's only one axon, though, that actually takes this information away. And if there's enough of a signal, the soma says, send the signal down, and it's going to go down these axons. Some of these are short, some of these are long. Some of these can be many feet. So the longest ones in your body are going to be like the axons with the vagus nerve or your sciatic nerve. That can be easily 3 feet long. You look at something like a vagus nerve in a horse, you're looking at something that could easily be 6 or 7 foot long for an axon. So these can be very, very long, and these make up some of the largest cells in the body, actually end up being nerve cells. And at the end of them, you're going to have what we call telodendrites. So dendrites up here take in the information. Information exits through the telodendrite, and at the very end of the telodendrite, these things branch off. Once again, 20 or 30 of these branch off, and they have synaptic knobs, and these synaptic knobs become very important as far as releasing neurochemicals. They don't actually touch, none of these things, no dendrite or no synaptic knob actually touches another cell. They take chemical messages from other cells and actually transmit them and turn them into, actually the dendrites take in chemical signals from other cells, and they're going to actually turn them into an electrical impulse, and that electrical impulse is going to actually tell chemicals to be released from these synaptic knobs. So the dendrite receives stimulus from other cells in the body, and depending on what these cells do, it can be receptors for heat, cold, touch, pressure, stretch, other physical changes. I mean, there's a multitude of different things that they can detect. And they're short in numbers and they're branched, and they have many different areas that they connect to and get information from. You have the center right here, the cell body of the soma, and then from there you're going to have the axon. There's only one axon, and the axon's actually going to take the information that the dendrites have picked up, the soma's passed on, and it's going to actually send it through down to these telodendrites. And some cells are not myelinated. These are very short cells. These are things like those Panacea corpuscles and Meissner corpuscles in the cells, but in the skin. But these are all going to actually sit there and hook up to a larger nerve that's going to have all these dendrites. So one of these axons right here might actually hook up to, say, 30 or 40 Meissner's corpuscles. They're going to take that information, transmit it down this axon, and actually take the information from there probably all the way up to the spinal cord. And then from there, you're going to have these little synapses that are going to take place, and it's going to communicate with multiple nerves in the spinal cord about what's going on. And so in a very long cell like that that's myelinated, you're going to have hundreds of these neuroglial cells that are going to sit there and support that very long axon. There's only one nucleus in a nerve, and so to actually support all the rest of the structure going On a very long cell like this, you actually have to have these other cells supporting that actually keep it alive. One of the things about myelination is it makes it to where the conduction of the impulse is 10 times faster with a myelinated nerve compared to an unmyelinated nerve. So if your nerves were not myelinated, it would take 10 times longer for your body to receive information. If it's really short, like a Mesner's corpuscle or a Panacea corpuscle, these things are very short. It doesn't really make that much difference. But in a nerve that's now going from the tip of your toe all the way up to the spinal cord, it makes a lot of difference if it's 10 times faster. Depending on where we find these at, these myelinated cells actually make a sheath around the axon. They're laden with fat, so they kind of have a whitish appearance, these neuroglial cells. And they wrap around, they support the cell, and they also help out with conduction and make the nerve impulse run a lot faster. If it's found inside the central nervous system, these are cells or what's known as an oligodendrocyte. And the thing about an oligodendrocyte is these actually will have myelin that will cover 3, 4, 5, 6 different axons. What's important is it's just one cell that covers this many, and what it does is it cuts down on space. It makes it to where the nervous system, central nervous system, the brain and spinal cord fits where it does. Whereas if you had what's on the peripheral nervous system, which is Schwann cells, which is an individual cell that wraps around the axons, if you had Schwann cells in your central nervous system, your brain would be probably in spinal cord probably about 5 times larger than it is now to accommodate all those Schwann cells. But by having the oligodendrocyte cells actually sit there and cover 3, 4, 5, 6 cells at a time, it makes it to where it's a lot more compact and it fits in the space that's available. Multiple Schwann cells or the oligodendrocytes, they end up covering that full length of that axon. And in between where each one of these cells are at, so as you can see, this is one oligodendrocyte cell. There would be another one here. There's another one over here. And so you can kind of see that here's a Schwann cell, here's your Schwann cell. And you can see the way that they wrap themselves. They physically wrap around that axon. But in between them, we actually have what's called the node of Ranvier. So node of Ranvier. And this is very important because this is where electrical impulses are going to take place. And we'll see this with saltatory conduction, exactly how that works. When we look at the nervous system, we actually organize it in multiple different ways. But the first one we're going to organize it into is anatomical. And so anatomical ends up being either the central nervous system or the peripheral nervous system. And it's a very easy distinction to make right here. The central nervous system is going to be the brain and the spinal cord. That's it. What's contained within the cranium and what's contained within the vertebral column, that is the central nervous system. The peripheral nervous system is all the nerves that extend out from the spinal cord and out from the cranium. So like cranial nerves, when we get into those, a lot of people get confused. They kind of think that they're part of the central nervous system. They're not. They're peripheral. They arise from the cranium, since we call them cranial nerves, but they're actually part of the peripheral nervous system. They're not part of the central nervous system. We also have spinal nerves, and so these are nerves that actually originate from the spinal cord and go out into the peripheral part of the body. Once again, they're not part of the central nervous system. They're part of the peripheral nervous system. We call them spinal nerves because they arise from the spinal cord, but they go out into the peripheral part of the body. So central nervous system is everything that's housed, all the nerves, and your brain can house inside the cranium, and everything that's through the vertebral column of the spinal cord. Once it exits that and goes out into the body, then you're in the peripheral nervous system. When we look at direction of impulses on organization of the nervous system, we have the afferent nerves, and so these are ones that actually receive information or carry information up towards the brain. So these are going to be sensory. So they're going to take information from the peripheral nervous system, take it to the spinal cord, from the spinal cord up to the brain, and so these are always afferent. Efferent nerves, and E is for exit. So efferent nerves carry impulses away from the brain and the spinal cord, and they actually go work on motor neurons, and so they're actually responsible for movement. And so whenever it exits out, there's going to be something that it's going to do motor-wise. There's some type of motor reflex that's going to take place with skeletal muscle contracting or moving. Potentially it could be like a gland, smooth muscle squeezing it and causing a hormone to be released. Motility of the GI tract. So all these different things are efferent effects. So if it comes from the brain and exits out into the body, it's efferent. If it comes from the body and it enters up towards the spinal cord and the brain, it is afferent. We get down to function. There's automatic and somatic. This has nothing to do with anatomy whatsoever, or afferent or efferent. The difference here is the somatic nervous system is conscious and voluntary. It means that you can think about it and your body can do it. Whereas the autonomic nervous system ends up being automatic. You don't have to think about it. You breathe, you don't have to think about breathing. Your heart beats, you don't have to think about it beating. These are things that you don't have conscious control of, whereas the somatic system would be like moving your fingers, moving your arms, or me talking right now. It's all voluntary and under control. So when we look at neuron function, we have what we call resting state. This is when a neuron's not being stimulated. It's perfectly at rest. And it has a resting membrane potential. We've already talked about action potential, and so this is a difference in electrical charge. So the resting membrane potential is when sodium is outside the cell, potassium is inside. And it's this difference in distribution right here that causes charges. And so when it's in the resting state, you have sodium outside, potassium inside. When we go into depolarization, what's going to happen is that sodium is going to rush into the cell and potassium is going to rush out of the cell. And what we use to keep this balance in this resting state is going to be that sodium-potassium pump we've already talked about, where sodium is pumped out into the extracellular fluid and potassium is brought back into the intercellular fluid. And remember, this takes ATP and energy for it to occur. When depolarization ends up happening, what happens is sodium is allowed to rush in. You're going to have a change of electrons and electrical charge when this happens. After that rushes in, potassium is allowed to rush out through the potassium gates. Once again, you get another change in ions and electrons, and so this is what makes an electrical charge, and this is what's going to actually travel down that axon, is this electrical charge. And that's what's called an action potential, is that swapping of sodium and potassium. When we get to repolarization, it doesn't really show a good thing right here with the diagram, because it's showing potassium running out, and that's not repolarization. Repolarization, all that really is whenever sodium is pumped back out of the cell and potassium is pumped back into the cell. That's repolarization. So depolarization is the changing of sodium and potassium's locations. Repolarization is actually sodium going back outside the cell, potassium coming back in, and restoring the resting state. When we sit there and look at repolarization, and the resting state is restored right here with repolarization, one thing to understand is that we do have this all or nothing of action potential. So either it's going to fully depolarize or it's not. And so to make it fully depolarize, you have to have a sufficient stimulus to actually make it completely depolarize. And it's that all or nothing principle that we talked about with muscles. It's either going to all depolarize or it's not going to depolarize at all. And so the conduction of this action potential is going to spread that wave down through there, through those sodium channels, and it's going to cause that depolarization. And it's going to make that electrical impulse and go down through there. We have this refractory period, and we've divided it into two different parts. There's the absolute refractory period. This is during the time when sodium and potassium are in such a balance as to where there's too much sodium inside, too much potassium outside, and there's no way that you can actually get back to a state to where you can depolarize again. Something a little bit different with nerve cells is they have a relative refractory period. So usually an impulse can't be conducted again until everything's perfectly back in place, where sodium's totally out of the cell, potassium's totally in the cell. But with nerves, sometimes we have what we call a relative refractory period. Near the very end of repolarization, if there's enough change in the ion balance right there to where you can get another action potential because you have just enough sodium outside the cell and just enough potassium inside the cell, but they're not back to that full equilibrium at the resting state, it is possible for them to repolarize or depolarize again during this relative refractory period. If there's an extremely strong stimulus, it has to be an extremely strong stimulus to do this. Muscles aren't able to have this relative refractory period. You have to have everything perfectly back in balance. You know, you have to have 134 parts of sodium outside and 134 parts of potassium inside for a muscle cell to fire again. But a nerve wants to be at that 134 and 134 equation, but If say we get to like 120 outside and 120 inside, it can, with a strong enough stimulus during this relative refractory period, fire again. But it has to be a very large and strong one for that to happen. Saltatory conduction, this is how actually impulses are going to be passed along. And so at these nodes of Ranvier, what it is you have an action potential here. And then what it does and what makes it so fast is that this action potential actually skips over this myelinated cell right here, the Schwann cell, and goes to the next node of Ranvier where you have your next action potential, which jumps forward to the next action potential. And so what would happen without it, if you follow my cursor here, is if you didn't have the myelinated cells, it would have to go like this: 1 to 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 1. Well, we can jump all those 1 to 1 to 1s, and we can go 10 at a time. So 10, 10, 10, and so it makes it a lot faster when you're having to do that. It's about like playing Monopoly and rolling a 10 on the dice. You get a lot further around the board than you do if you're rolling twos. And so this is kind of the thing right here is, well, actually on Monopoly you can roll 12. And so, you get a lot further on the board when you do 12. And so that's kind of this principle right here. It's taking 12 spaces at a time than if you roll 2 on the dice where you can only go 2. So if you roll 2, you'd be here, but you roll 12, so you're over here. So it makes it much, much faster to get that impulse back and forth. Synaptic transmission, so between those synaptic knobs is what we call a synapse. And so it's the junction between two neurons or a neuron and another cell. And there's this little gap in between them called the synaptic cleft. And so the presynaptic neuron, the one that actually has the little synaptic knob on the end of it, when the electrical impulse gets to it, it's actually going to cause little packets of neurotransmitters that are right there at the very end of the synaptic knobs to actually go ahead and take those little vesicles with those chemicals in them, those neurotransmitters, and actually fuse with the plasma membrane or the cell membrane and actually open up and release those chemicals out into that synaptic cleft. And the postsynaptic neuron or the postsynaptic cell is going to actually contain receptors for those neurotransmitters. And so this is how signals are actually passed on by nerves. So at the end of it, it's those little branch structures we talked about on the presynaptic ends called telodendrites. Each one has a terminal bouton, or what we call a synaptic knob, and in there's little vesicles in those knobs that have the neurotransmitters. So when that electrical impulse happens, that vesicle fuses with that cellular membrane and actually releases that in the cleft. And so here's kind of a picture of that right here. So you can see axon goes into a telodendrite. You have the synaptic knob right here. They have these little vesicles. Let's say like this is acetylcholine. This is, say, a muscle over here. The electrical impulse happens. This vesicle is going to actually fuse with that plasma membrane or that cell membrane, and it's going to release the acetylcholine. And the acetylcholine is going to come across, and it's actually going to attach to receptors over here on this same muscle cell. And by actually attaching to these receptors, this is actually going to cause the muscle cell to actually contract. So this is the way that it actually works. Nerves don't actually touch one another or other cells. They actually get very close to the end, and these things are micron in distance. They're very, very tiny in distance apart, but it's close enough to where those chemicals get there, but they don't physically touch. And these chemicals are released right into this junction right here. And this is what causes them, whatever it is, muscle to contract, another nerve to fire. So when we look at neurotransmitters, we have excitatory and inhibitory. So excitatory is going to cause an influx of sodium at the postsynaptic knob. It's going to cause some type of action potential to happen on the other side. Inhibitory actually sits there and makes it to where another action potential or another threshold is not going to be able to take place. So when we start looking at it, there's one that ends up being both inhibitory and excitatory, and that's acetylcholine. And so acetylcholine, depending on where it's at, is what it's going to do. So if acetylcholine is in muscle, it's excitatory. It causes your muscles to contract. Funny thing is if it's at the SA node of the heart, acetylcholine actually makes your heart rate slow down. So it's excitatory for skeletal muscle, but it ends up being inhibitory for cardiac muscle. We have a whole group of excitatory chemicals, neurotransmitters, called the catecholamines. These are going to be things like norepinephrine, epinephrine, and dopamine are the main three that we're going to talk about. And so all of these actually end up being excitatory, and they cause some type of neurotransmission or transmission to muscles or something to go on out in the body, and it actually excites them. The one that is strictly inhibitory ends up being gamma aminobutyric acid, or GABA for short. Another one is glycerin, but the one that we'll actually look at the most is GABA. So GABA is very important because it's the inhibitory one. It stops action potentials. And so while these catecholamines get very excited, GABA steps in and actually kind of slows them down. And the importance of GABA is without GABA, all the catecholamines take over, and you get disorganized patterns. You get things like seizures. Can occur if you don't have enough GABA to actually inhibit the catecholamines. So it keeps everything in balance. GABA allows enough excitement for things to get done, but not so much that the pattern gets disorganized and you start having seizures and paralysis and other problems. This gets really important when we start talking about things like certain dewormers. We have the group of avermectins, and they actually work on parasites by inhibiting GABA. And when we get into the blood-brain barrier, we'll find out why it's important to know about certain drugs and their effect on the nervous system and how they would affect things like catecholamines or GABA. Because if you start interfering with these, you can actually interfere with the brain and its function and cause either severe illness or death. So stopping neurotransmitters, and they also call it recycling neurotransmitters, this actually occurs when we have something, usually it ends with ACE. So any enzyme that ends with ACE means to break down or to block another enzyme. So acetylcholinesterase is actually found on the postsynaptic membranes. And so it's going to be found on muscle cells or other nerves that after acetylcholine's been attached, they're actually going to release the acetylcholinesterase. And its whole job is to actually go out there and break down the acetylcholine. And by breaking down the acetylcholine, it actually stops the contraction. So acetylcholine is released, acetylcholinesterase comes out, breaks down that acetylcholine. If you want the muscle to contract again, you release more acetylcholine. And so this cycle goes on to where acetylcholinesterase comes back and breaks it down. Monoamine oxidase, or MAO, ends up breaking down norepinephrine. Some people have a problem of not having enough, so we have MAOIs, monoamine oxidase inhibitors. We use this a lot for depression because norepinephrine has a lot to do in the brain with regulating moods. And so if we use an MAOI, we allow the amount of norepinephrine in the brain to actually increase. And that's what some people need. There's also what they call catechol-O-methyltransferase, which also breaks down norepinephrine. And this is usually found not in the nervous system, but more out in the body. And these are the main ones that we look at right here. And there's some important things like with acetylcholinesterase. One of the things that happens like with tetanus or lockjaw is actually the fact that acetylcholinesterase is inhibited from being released. And so what makes the muscle so rigid from the tetanus toxin, if you happen to get tetanus, is this Clostridium tetani that produces this toxin. When the bacteria dies, they release this toxin throughout the body into the bloodstream, and actually it blocks acetylcholinesterase. And so it means that acetylcholine just stays on the muscles and it makes all the muscles rigid. And that's what it makes people's jaws clamp down and they get lockjaw. And don't forget that the diaphragm is a skeletal muscle too. And what usually gets these animals, if they happen to get tetanus, is the fact that it actually sits there and makes the whole diaphragm rigid and they stop being able to breathe. So these things end up being very important in a lot of the different diseases we see. We're going to talk about the central nervous system, and the central nervous system is broken down into two parts: the brain, which is this part right here, and the spinal cord. We break the brain actually down into four distinct areas. We have the cerebrum, or what's also known as the cerebral cortex, where voluntary thoughts, sight, smell, all that's recorded, voluntary actions. We have the cerebellum, which is important for coordinated muscle movement and balance. We have the diencephalon, which lets different parts of the brain communicate with each other. It's also important for releasing hormones and sensing things like temperature, different noxious substances in the body, and getting responses for things like vomiting or producing a fever or releasing hormones needed for the body to function normally. And then we actually have what they call the brainstem, and it's further broken up into three different parts: the midbrain, the pons, and the medulla. And so this right here is what's known as the brainstem area right here. And so these are the four parts of the brain. And then the spinal cord, the brainstem actually attaches to, and it's going to travel down the vertebral body. And we actually break it into different sections. Cervical part of the spinal cord for the cervical vertebra, thoracic part for the thoracic vertebra, lumbar for the lumbar vertebra, and then it ends in the sacral area for the sacral vertebra. Connection between the spinal cord and the brain is actually going to be the brainstem right here, and specifically the medulla oblongata portion that we'll get into. So when we look at the cerebrum is made of gray matter. Gray matter is soma. It is cell bodies. White matter ends up being axons. So when we look at the nervous system and the central nervous system, you can look at the color and figure out, am I looking at cell bodies like somas, or am I looking at a bundle of axons by looking at whether it's gray or it's white in composition. And the thing about the cerebrum is that it is made up of, majority is gray matter. It's where there's a lot of somas at. There's a lot of thought, a lot of higher processes that go on. Deep inside the cerebrum, you actually start finding white matter, specifically at the areas called the corpus callosum, which actually allows the right half of the brain to speak to the left half of the brain, and also communicates down into the diencephalon. And the cerebrum is all your higher order behaviors, learning, intelligence, awareness, thought, emotions, all of these different things come here. Also your higher senses like smell, taste, sight, these are all interpreted within the cerebrum itself. On the outside of the cerebrum, we have what they call gyrus or gyri. If they're plural, these are ray spots. And then sulcus or sulci, and these are depressed. And so by putting these together, it's what we call folding. And the higher level of intelligence brains are the ones that are highly folded like ours. Ours is not actually the largest brain. There's actually whales and elephants and some animals with some larger brains. But they don't have near the folding, which actually increases surface area. So remember, when you look at the brain, the raised section in this folding through here is going to be known as the gyruses. These dark lines meaning valleys, these are the sulci or the sulcus. You also have a right and left hemisphere, and you have the longitudinal fissure that actually separates your brain into the right side and the left side. When we start looking at the cerebellum, it's back here. Its important features ends up being it has a lot to do with proprioception, knowing where your body's at, your hand and your feet are at all times. It plays a large role in balance and equilibrium. And then it makes your muscle movements very smooth and coordinated. So a lot of people that have problems with things like Parkinson's, they actually come from the cerebellum because the muscle movement is no longer coordinated when you have something going wrong with the cerebellum. The diencephalon, which is deeper down the brain, it's important for keeping a lot of your hormones and a lot of your regulatory systems going in the body. It's the area that helps to sense things like fevers and temperature, and it can also help to sense different noxious substances in the blood and can actually initiate things like vomiting. You have the thalamus, which is a relay station and is actually very important for regulating these sensory inputs. And it plays a role in vomiting, plays a role in body temperature, plays an overall role in homeostasis. The hypothalamus is a very important part because this is the interface between the brain and the endocrine system. And so the hypothalamus actually controls a lot of the endocrine system and what hormones are going to be released. At the base of the hypothalamus is going to be the pituitary gland. And the pituitary gland is actually not a part of the brain. We have it here with the diencephalon. It's located next to the diencephalon, but the pituitary is actually an endocrine gland, and it's the master endocrine gland. And it takes all of its orders from the hypothalamus. Finally, you get to the brainstem. This is the connection between the brain and the spinal cord, and you have the medulla oblongata, the pons, and the midbrain. And so each one of these is very important for running a certain part of the autonomic nervous system. This is the part of the brain that you don't have to think about, that does everything just automatically and on its own. When we actually start looking at it, things like the medulla oblongata ends up becoming very important because this is where your respiratory centers are at, and the centers to keep your heart beating is in the medulla oblongata. In here, you're also going to find a lot of these different baroreceptors and chemoreceptors inside the brainstem that's going to sense things like oxygen, carbon dioxide, your pH, noxious chemicals, and so it's going to make certain controls to your body based on it. And it helps to keep the digestive system and everything working in the brainstem. So it keeps all those basic rudimentary autonomic functions going. Meninges, these are a tissue that actually covers and surrounds the central nervous system. So these are actually going to cover the brain and the spinal cord. They contain blood vessels. There's some fat in there, so they're there for cushioning and stuff. These blood vessels can help actually with supply nutrients to the outer surface of the brain and spinal cord. The fluid in there is very important for cushioning of the brain. If you do not have it in your spinal cord, just simple movements of your head would actually cause severe bruising to your brain. So there has to be that fluid. And then there's some also fat in there also for cushioning of the brain. This fluid also is very important because it also carries a lot of white blood cells that are looking for anything that's trying to attack your central nervous system. Central nervous system controls everything going on in the body and gives the inputs for everything going on, so it's very important that it's protected. And that cerebrospinal fluid that's around these meninges does that. So when we actually look at it, there's 3 different layers. So out here is the bone, the skull. And this very outer layer right here that you see in yellow is known as the dura mater. And the dura mater is very important because it's a very tough fibrous protector of the central nervous system. And so it's made of this very dense, irregular connective tissue. It's very thick, very tough to actually cut through. And it actually carries blood vessels, some arteries that are going to go down to the brain. There's larger arteries out here on the surface of it, and some smaller ones go down. And then you have a lot of venous blood return that actually comes back up to the dura mater for blood to be taken away. Laying on top of the brain and the spinal cord is actually something known as pia mater. And this is very thick and durable. This is very fine and lacy, this dura mater. And it just lays right where this black line is. It's just this very fine, lacy-like material. In between the dura mater and the pia mater is actually what's known as the arachnoid space. And the arachnoid space is this web of connective tissue and small, fine blood vessels that run through here. A lot of these are veins, some of these are arteries. But they run in between here, and what actually circulates through here is a lot of your cerebrospinal fluid. So your cerebrospinal fluid is actually found in this arachnoid space. Some importance about this is the cerebrospinal fluid should be clear when we look at it. So we don't want to see things like it's cloudy in appearance. It shouldn't be yellow in appearance. It shouldn't have any blood in it. So those are all signs that there's something wrong if we find that in the cerebrospinal fluid. Also in this space, if anybody's ever heard of a subarachnoid bleed or a subepidural bleed, this is actually in the arachnoid space, and so this is when venous blood actually starts to fill in. And when that happens, it actually puts pressure on the brain. There's also something known as an epidural bleed, and this is between the bone and the dura mater. And these things are especially serious because the things with epidural bleeds is subdural hematomas can be fatal, but they're less so. Epidural hematomas, if they're untreated, are almost always fatal because this arterial blood actually starts pushing the dura mater down. And then over time, with any type of pressure bleed, if you get enough pressure from either fluid or blood surrounding the brain, there's only one exit for the brain, and that's through the foramen magnum, if you remember that one hole that the spinal cord exits from the brainstem. The problem is that it will actually push the brain and try to force it through that foramen magnum. When that happens, we call that herniation of the brain. And when herniation of the brain happens, the medulla oblongata, which is important for your heart rate and your respirations, is actually forced into the vertebral column, and the vertebral column's too small for it, and it puts too much pressure on the medulla oblongata and actually damages those cells. And so any animal that has herniation of the brain from too much pressure here, from too much fluid or too much blood in the arachnoid space or between the dura mater and the skull, if they actually herniate, death is going to actually occur. So the cerebrospinal fluid is going to be actually made in the brain between the diencephalon and the cerebrum is an area known as the ventricles, and so these actually produce it. There's actually going to be a canal that this fluid actually goes down the center of the spinal cord. It's actually going to exit back around the sacral area, and then it flows back up to the brain where it's actually reabsorbed again. And said it does functioning. The functions include cushioning and has a lot of white blood cells in there. Not like a ton of them, but more than enough of them to actually take care of any type of foreign invader or anything that's going wrong inside of that space. It's also an important area where the body actually looks for carbon dioxide and determines respiration. The body also looks at the cerebrospinal fluid, and if it notices any toxins there, it can actually initiate vomiting based off of what's in the cerebrospinal fluid. The blood-brain barrier, this has nothing to do with the meninges. And so this is where we talked about very earlier about those catecholamines and the GABA, those excitatory and those inhibitory chemicals. In mammals and avian, we have a blood-brain barrier. Reptiles, amphibians, and fish do not. So it's very important to know what type of drugs that we're using because some drugs have an effect on the nervous system and you wouldn't even think it, like a lot of our dewormers do that we learn about in pharmacology. So if any of these drugs or chemicals actually enter the brain, they can have a very negative impact on the brain. They can cause death through seizures or through inhibiting function of the brain and making it actually shut down. And what mammals and birds have is this thing called a blood-brain barrier. And so in every capillary there's a fenestration. If you remember us looking at the circulatory system, when we looked at capillaries, there's little holes that allow things to come in and out of the capillary. Oxygen to very easily diffuse out, and carbon dioxide comes in very easily, but it also allows proteins and other things to cross the nutrients out into the cells and allows other nutrients, or other waste products to come back from the cells and enter back into the circulation. And what happens with the blood-brain barrier is actually on those capillary fenestrations are closed off by those neuroglial cells, those oligodendrocyte cells. They actually sit there and actually block off all those fenestrations in the capillary. So they keep things from freely passing to the brain. And once again, mammals and birds have this, and avians. Reptiles, fish, and amphibians totally lack this. And so in those animals that lack it, a lot of our different things like an avermectin dewormer, like ivermectin, that we can safely give to a bird or a mammal would actually kill something like a snake or a fish or a frog. And that's because they lack this blood-brain barrier. And what the blood-brain barrier does is it's very specific. It does not let hydrophilic things in, things that like water, because these neuroglial cells are packed with lipids. There's a lot of fats there. So something has to be fat soluble to actually cross in. An example of this is looking at antibiotics. So when we look at penicillin, penicillin is very hydrophilic. It loves water. It does not cross into the brain easily. So if we're trying to use it to treat something like a bacterial meningitis, penicillin is a very poor choice because it does not actually cross over into the brain that easily. On the other hand, our sulfa antibiotics, they're very hydrophobic. They're lipophilic. They like lipids. They hate water. And they very easily cross into the blood-brain barrier. So if we had something like a bacterial meningitis, we like to treat it using sulfas. And it's because of this blood-brain barrier that it does this. And so understanding that principle is very important in pharmacology. It's very important to understand that it's there to protect our brain from things that would be noxious to it, and our spinal cord too. Things that would be noxious to those neurons that would actually disrupt them and potentially cause death to us. Next we have the cranial nerves. Cranial nerves are always going to be used for Roman numerals, and they're 1 through 12. Each cranial nerve hooks up to a part of the brain and is going to either do one of three functions. It's either going to be sensory, so it's going to sense something in the environment. It's going to be motor, so it's going to tell the body something to do. Or it's going to be both. It's going to be both sensory and motor. And so when we look through it, our cranial nerve number 1 is olfactory. It is strictly sensory, and it's important for the sense of smell. Our cranial nerve number 2 is the optic. It's sensory. It's for vision. Cranial nerve 3 is oculomotor, and it's important because it actually allows for change in pupillary size, focusing the lens, and movement of eye muscles. It actually helps. The oculomotor actually goes to the muscles that go to the back of the eye and actually helps to move the eye and actually retract the eye. Next you have the trochlear nerve, which is cranial nerve 4, which is motor, and it allows movement of the eye from side to side and up and down, and it plays a role in that. Cranial nerve number 5 is the trigeminal. It's both sensory and motor. And so it takes sensation from the mouth and around the head from things like chewing, the teeth, and certain areas of the head it actually takes sensations from. And then it's important for things like mastication and moving and chewing with the mouth. Cranial nerve 6 actually is the abducens nerve, which is motor, and once again it plays a role in eye movement, moving your eyes around. Cranial nerve 7 is the facial nerve, and it's both sensory and motor, and so this one actually goes out to your lips and around your nose and stuff, senses a lot of things on your skin, your scalp. It's important for things like salivation, making tears, taste, and it's actually important for actually blinking your lower eyelids, and that's what the facial nerve does. The vestibular cochlear nerve is sensory, and it actually plays a role in both hearing and balance, so it actually does both. The glossopharyngeal is both sensory and motor, and it actually goes to your pharynx and to the base of your tongue, and it plays a role in things like gag reflexes, controls the motor muscles back in the back of your pharynx and at the base of your tongue. Cranial nerve 10 is the vagus, and it actually leaves everything else we've talked about has been up in the head and the skull area, but the vagus nerve actually leaves and actually goes to your thorax and your abdominal area. It's a very long nerve, and it actually is both sensory and motor. And it plays a role with your gastrointestinal tract, your respiratory tree, motor to your larynx and your pharynx, your parasympathetic system. All these different things are controlled by the vagus nerve. Cranial nerve 11 is the accessory, and so, or spinal accessory is the proper name for it. And spinal accessory controls the muscles that actually move your neck, your head up on top of your neck. And so that's what the spinal accessory does. And then lastly, there's the hypoglossal nerve, and this actually controls the movement of your tongue. So if you lose control of your hypoglossal nerve, this is when you bite your tongue. So it does all the rostral portion of your tongue. We have some different sayings and mnemonics, and there's actually in the book, there's a mnemonic on page 328, table 13-2, that goes on old Olympus towering tops, a fine vocal German viewed some hops, or you'll hear me say, on old Olympus towering tops, a fat vested German viewed some hops. You can use either one, but this is to help you know when you say the O's on old Olympus. That means olfactory, optic, and oculomotor. Towering tops is trochlear and trigeminal. A is for abducens. Fat or fine, either one you want to use, is for facial. Vocal or vested is for vestibular cochlear. I like vested because it goes with vestibular cochlear myself. German is, of course, for glossopharyngeal. View is vagus. Some is spinal accessory, and hops is hypoglossal. And so using a mnemonic like this can help you actually keep which nerve is in order. If you go through and you want to know the third one, you say on old Olympus, then you're like, third one, you know Olympus is oculomotor, then you know that's your third cranial nerve. So that's kind of a way to help you put them together and have a mnemonic to where you can say them and try to figure them out. A couple of things to remember is trigeminal is always number 5, olfactory is number 1, and optic is number 2. Facial is always number 7, so remember that, facial 7. Vagus is a 10, so always remember that. So the thing that we learned in vet school was that going to Vegas on vacation is 10 out of 10 for vacation spots, so Vegas is 10. I don't know if it is, I haven't been there personally, but that's just the saying that we use. And then, so by learning these in certain numbers, it can kind of help you out to know that trigeminal is 9, facial is 7, and vagus is 10. You can kind of fill in the gaps from there. The numbering system, the number 1, the olfactory, is the most rostral on the brain. And then number 12, hypoglossal, this one actually comes off at the base of the medulla oblongata. So they are numbered sequentially. So, you know, the first cranial nerve you come to is the olfactory. The second one, if you're coming rostrally, the second one is going to be the optic nerve, and we'll see the optic chiasm, followed by the oculomotor, trochlear, trigeminal, abducens, facial. So they all come off in order from rostral to caudal. Also, when we start looking at things like whether they're sensory or motor, there's actually a saying here. I learned it as 7 sailors made merry, but my brother said, bad business, my man. And here they have 6 sailors made merry, but my brother said, bad business, my man. And so what this does is anywhere you have a word that begins with S, so you can sit there and write down on your test, your cranial nerves, 1 through 12, and then you're going to have to repeat whether they're sensory, motor, or both. And so on that mnemonic, 6 sailors made merry, but my brother said, bad business, my man. If the word begins with an S, then it's sensory. So 6 is sensory for olfactory. Sailors, which is an S, is sensory for optic. Made is an M for motor, for oculomotor. Merry is an M for the trochlear, which is motor. But, that's the trigeminal, that's both. My, which is abducent, which is motor. Brother, which is both for facial. Said, which is sensory for vestibular cochlear. Bad, both for glossopharyngeal. Business, which is once again a B for both for the vagus. And then my, which is motor for spinal accessory. Man for glossopharyngeal for motor. That kind of helps you line it out when you come down to a test, which one's sensory, which one is motor, and which ones are both. And you will have to actually do that on your lab test. You will have to list these things out. You have to list out cranial nerves. I'll put the 1 through 12 for you. You have to tell me which nerve it is and if it's motor, sensory, or both. When we look at the spinal cord, we're actually going to have the dorsal portion and the ventral portion. We have what we call the dorsal horn in the center and the ventral horn. And this is actually gray matter. This is actually soma, and this is actually in the area called the medulla, the very middle of the spinal cord. So when we actually look at the dorsal horn right here, we're going to have coming in what we call a dorsal nerve root. There's actually going to be a dorsal root ganglion area here where a bunch of these peripheral nerves are actually going to meet up. They're actually going to meet up with other nerves that are going to pass information here into the dorsal horn. And this is always going to be afferent. So this is always going to take information in here into the dorsal horn and the medulla. And then that information is going to be processed. And then information is going to leave out through the ventral horn to efferent nerves. So the dorsal side is always afferent, bringing information in. The ventral side is always efferent. It's always sending motor information back out. In the very center of the medulla is actually what they call the central canal, and this is where cerebrospinal fluid is actually going to flow down the center of the spinal cord. The outer portion is the cortex, and this is all white matter. And so all this white matter out here, this is all axons moving through here. So the very center is the medulla, it's gray matter. The outside of it is the cortex. This is all going to be white matter. And the more central the white matter is in the cortex, the more cranial those nerves are going to exit. And the ones that are more peripheral out here, the more caudal they're going to exit. So this is looking at an area, say, at the first cervical junction. This white matter really close to these horns right here, this medulla, is going to go to your thoracic limb, whereas the ones out here on the periphery, these are actually going to go to your legs and to your toes. And that's the way that they're kind of arranged. And this is important when we actually start looking at disease processes to understand, like when we have pressure on the spinal cord or injury to the spinal cord, why we sometimes lose motor and sensation to, say, the hind legs on an animal and not necessarily to the forelimbs. So kind of talk about that dorsal ganglia right here and the dorsal horn, and the fact they're always going to be afferent, they're always going to be bringing information in. And if you damage these roots right here, and this happens sometimes, they can be torn or damaged, or you can have a tumor here, you stop getting sensory information into the spinal cord. And this can be information such as touch, sensation, proprioception, pain, all these different things are going to be brought in. The ventral horns are always going to be motor. So if you ever have any type of injury down here to the ventral horns, then and the ventral nerve root, then we're going to actually be affecting motors. So you might be able to feel something, but you might not be able to move those muscles to do something about it. So the dorsal is always going to be bringing impulses in and afferent. The ventral is always going to be sending information out for motor or efferent. When we look at the nervous system, we get into things like the autonomic nervous system, and this is one of those subdivisions from the somatic that we look at separately. And so the autonomic nervous system actually controls everything at an automatic level. It takes care of all the subconscious things, the things that you don't need to think about. Because if you did, you would probably die, like keeping your heart beating, keeping respirations going, keeping the endocrine system functioning, keeping the digestive system functioning, keeping the reproductive system, the urinary system. All those different things are actually going to be controlled through the autonomic nervous system. And we further break it down into the sympathetic nervous system and the parasympathetic nervous system. And the sympathetic nervous system is fight or flight. It's about the things to keep you alive. The parasympathetic nervous system is what we call rest and digest. It's about all the things you do when you're relaxed. When we actually look at it, the sympathetic nervous system actually comes off in the thoracic and the lumbar region. So all the nerves that affect it come off in the thoracic and lumbar region. And we'll actually see in the next couple of slides, there's actually something here called the sympathetic chain, where they actually have ganglion that actually communicate with each other when they come off. And this is really important because if you have, say, a severing of the spine right here between the thorax and the lumbar area down here, some of the functions that take place on the sympathetic side further down can be temporarily disrupted by this break in the spinal cord. But eventually, because of this chain, they're going to actually get back their actual function again. So any breakage there is temporary. It's never permanent. Parasympathetic's a little bit different. It comes off of the vagus nerve, and it's one long nerve that's going to run down to the intestinal tract and to the lungs and into the heart. And so it's going to have branches that come off from there. And so it's one long one that doesn't actually sit in the central nervous system, but runs right along beside it. And then going to the bladder and the rectum and part of the large intestine, you actually have the parasympathetic that's actually going to come off of the sacral. So when we look at the parasympathetic, it comes off the vagus nerve and different cranial nerves. In the sacral region, all the sympathetic inputs actually come off of the thoracic and the lumbar. When we end up looking at the sympathetic nervous system, one thing we're going to notice is what we call the preganglionic neurons. So the ones coming off the spinal cord are going to be relatively short. They're going to synapse, and they're going to synapse along that ganglion chain. And so they're actually going to communicate along that sympathetic chain, as we call it. And then those postganglionic neurons are going to be extremely long. They're going to go from the spinal cord down to their targeted organs or the targeted tissues. And so this is what it's kind of talking about on this slide right here, is that it goes to that chain, short little roots that are preganglionic, and then the postganglionic are very long, and they actually go to wherever they're needing to go to, and they cause the effect. Parasympathetic's actually a little bit different. Its preganglionic are quite long. The postganglionic are going to be found in the targeted organs, and the postganglionic are awfully short. But remember, a lot of these are coming off of the vagus nerve. So the vagus nerve is very long, and the vagus nerve is that preganglionic section. So these are very long, and then the postganglionic section is going to be very short inside the targeted organs. We kind of look at the effects between them, and in your book right here where it says no significant effect, I don't really like that word. I'd rather say returns to normal. But we're going to look at things. So underneath the sympathetic nervous system, the heart rate's going to increase, and the force of contraction of the heart is going to increase. So it increases both rate and contraction. The parasympathetic causes the heart rate to decrease and fall back to normal, and it actually causes the contraction to go back to normal. It's not an insignificant effect. What it does is it returns contractions to normal. And that's actually pretty significant to me, that it actually goes back to normal. So sympathetic increases heart rate and heart contractility. Parasympathetic decreases heart rate and returns contractility back to normal. Looking at the bronchioles, the sympathetic is going to dilate them, increase their size. The parasympathetic is going to let them constrict or actually return back to their normal size. Pupil diameter is going to increase with sympathetic. If you're in a fight or flight situation, you want to see everything, so your pupils greatly dilate. And parasympathetic constricts them. It lets them go back to a normal size that the light determines their size. So it actually sits there and brings them back down in size to what would be normal for the light outside. Blood flow to the gastrointestinal tract. and causing of secretions and blood flow to things like the reproductive tract, the gastrointestinal tract, different things like that. Sympathetic input is going to decrease that. Parasympathetic puts it at normal. So it actually allows for normal or increases, but really just lets everything, gastrointestinal motility go back to normal, secretions like saliva go back to normal, blood flow to a lot of different organs in the reproductive tract and digestive tract and urinary system goes back to normal. Diameter of blood vessels in the skin is going to get smaller. It's going to constrict with sympathetic input. And it's going to return back to normal with parasympathetic. So once again, it's not a no significant effect. It actually goes back to normal, which is pretty significant. But it returns to normal. So you can just put in your book, returns to normal right there. Diameter of blood vessels to muscle. Sympathetic is going to increase that. Once again, we're looking at fight or flight. So to be able to run or to be able to fight, you have to have your skeletal muscles with peak blood flow. So it increases blood flow not only to the brain and the lungs and the heart, but it increases it to the muscles. What a parasympathetic does is it returns those blood vessels back to normal size. Once again, it's not a no significant effect. It returns it back to a normal size for function. So it takes a lot of that excess blood that was being shunted there and returns it back to the things like the gastrointestinal tract. And then blood flow to the kidneys. Sympathetic decreases this, and this becomes important because when you're normally resting, your kidneys use 25% of the oxygen you take in, which is about the same amount that your nervous system and your brain is taking in right now, is about 25%. So one way the body can decrease and conserve on oxygen to send to the brain and more to the brain and the heart and the lungs and to the skeletal muscles to decrease the amount of blood going to the kidneys. And so it does this by vasoconstricting the kidneys. So that's one of the sympathetic effects, is it actually decreases blood flow to the kidneys in the sympathetic system. Parasympathetic, once again, it's not an insignificant effect. It's a very significant thing, is it allows normal blood flow back. So when they say no significant effect here, it is a significant effect because it actually allows for normal. So it doesn't increase it, but it allows it from being decreased to back to normal, which is very important. So when we start looking at neurotransmitters that come off of the sympathetic and the parasympathetic system, we're going to look at the sympathetic ones first. And the main one that we have here is, we call them adrenergic neurons. And these are actually going to release norepinephrine. And so when we look out in the body, we're going to have some that are actually going to release just norepinephrine out to certain targeted tissues, and they're going to do certain jobs. Other adrenergic neurons are actually going to release norepinephrine, and they're going to cause other chemicals to be released. And these other chemicals can be things such as epinephrine itself, or we can actually get over into what we call our adrenergic receptors. And so these adrenergic receptors, norepinephrine or epinephrine, will actually cause certain things to be released. So our first one is actually what we call alpha-1 adrenergic receptors. And so the effects of norepinephrine and epinephrine on these things actually causes things like vasoconstriction of the skin and the GI tract and the kidney. So your alpha-1 receptors do this. They don't have alpha-2 on here, but alpha-2 is actually in the brain and spinal cord. And so is alpha-1. Alpha-1 actually causes a heightened amount of excitement. Alpha-2 we actually can use as a sedative, but what it actually does is it reduces the brain's perception of things such as pain. And they don't have it on the slide, but alpha-2 is just as important. So alpha-1 constricts blood flow to the skin and the GI tract and the kidneys. Alpha-2 is going to sit there, and it's actually going to play an important role in actually making it to where you don't perceive pain. So alpha-1 also goes along with heightening awareness. Alpha-2 kind of counteracts any sensations of pain. Beta-1 actually increases heart rate and is what's important with increasing force of contraction. With people that have certain heart conditions, we have beta blockers, and it actually blocks beta-1 input. So it actually kind of slows their heart rate down and decreases their blood pressure. And then beta-2 is very important because it causes bronchodilation. So most of your asthma medications actually work on beta-2. It actually causes that bronchodilation to occur. And so this alpha-1, alpha-2, beta-1, beta-2, these adrenergic receptors here are very important as far as regulating sympathetic responses and the job that they're going to do as far as in the nervous system with alpha-1 and alpha-2. Out in the skin, the GI tract, and the kidneys with alpha 1 with vasoconstriction, beta 1 increasing heart rate and force of contraction, and beta 2 actually causing bronchodilation. When we look at the parasympathetic nervous system, we have cholinergic receptors, and these are going to be acetylcholine. So you can have straight acetylcholine, like what works on the SA node and kind of slows down the heart rate. And then we actually look at specific ones. So in a lot of our out in the periphery of the muscles and up in the brain, we have what they call the nicotinic acetylcholine receptors, and we call them nicotinic acetylcholine receptors because nicotine influences them. So the body makes a small amount of these nicotinic acetylcholine receptors, and so it actually has a work between the parasympathetic and a lot of your different motor neurons and a lot of your different muscles, both smooth muscles and skeletal muscles throughout the body. The muscarinic acetylcholine receptors is named after muscarine, which is a poison, and its effects are actually on different tissues. So things like the liver, the kidneys, and the lungs. And this is where the muscarinic acetylcholine receptors are actually going to have an effect on. And when they're stimulated, they actually cause parasympathetic effects in those areas. So we look at the nicotinic and the muscarinic. There's different areas. You're going to find the nicotinic a lot more out around with muscles and have a lot more of a motor function. The muscarinic you're going to find a lot more in specific organs, and they're going to have an effect on those organs. And they all have an influence on influencing the parasympathetic system and getting it to kick in for that rest and digest. And so you can kind of see the autonomic nervous system. Sympathetics kicked in. You have norepinephrine. Norepinephrine also works on these adrenergic receptors, alpha 1, alpha 2, beta 1, beta 2, and causes the chemicals to be released to stimulate these. Parasympathetic, you have straight acetylcholine, or you have the or the cholinergic receptors, and you have the nicotinic and muscarinic acetylcholines that are released that are actually going to stimulate those. Lastly, we get to reflexes, and reflexes are very important. When we start looking at reflexes, we have 4 different types. We have somatic reflexes. These are actually going to involve skeletal muscle contraction. Autonomic reflexes, these are going to be like smooth muscle, cardiac muscle, and endocrine glands. We further break down somatic reflexes of the body into contralateral reflexes. So a reflex on one side of the body causes an opposite reflex on the opposite side of the body. And ipsilateral reflexes, so a reflex, say, in the leg causes a reflex also in the arm. When we look at a reflex arc, this is actually some type of sensory reception to something noxious in the environment. Something has come along that is not normal and is going to cause a reflex to occur. And so there's something that's potentially painful or harmful, and a nerve impulse is sent from the peripheral nervous system, and it actually joins in either where a cranial nerve meets the brain at, or it's going to come from where a spinal nerve comes into the spinal cord from the peripheral nervous system. And it's going to input into the central nervous system. And something's going to be detected. And with a reflex, what ends up happening is when that thing is detected, the body's going to have some type of withdrawal or movement away from it. And this is not going to be conscious. This is a reflex. This is something the body does automatically in order to protect itself. So here's a good thing with a reflex that you can see is something is coming at your face really fast. What is your reflex going to be? Your optic nerve sits there and senses it, tells your brain, hey, there's a baseball coming towards your head. And so your reflex is going to be, as this thing gets close, is to all at once, it's going to blink, and you're going to potentially duck. And then your brain's going to register there was a baseball near my head. So the body sees it, notices the danger, reacts to it, and then you actually have the thought about it. Or you step on something sharp. As soon as you step on it, your foot withdraws from it, and then that's the reflex, and then your brain senses the pain. And so these are protective mechanisms when we start looking at reflexes and reflex arcs. The most simple one that we study is the monosynaptic reflex arc. And this is the one where the doctor taps you on your knee and you kick your knee out. And so this is actually pressure on a specific muscle. And so this pressure says, hey, this muscle's overstretching. I need to protect it. And so the thing when you kick your leg out, it keeps the muscle from stretching any further and actually tightens it up. And so there's only one synapse that goes on. The pressure arc goes in here. It ends up coming up here to the spinal cord. It's going to go into one area specifically in the spinal cord. And it's going to come in here through the dorsal horn. And it's going to have a couple of synapses that go on right here. And so these synapses are actually going to sit there and send the reflex back out and tell the leg to kick. And then it's actually going to send a signal up to the brain and says, hey, that was stretched and I just told everything to kick. And then you realize it, because if we're waiting for the brain to do it, it would take too long. By the time that a reflex happens and your brain processes it, everything's over with very fast. If your brain had to process it and tell your leg then what to do, it could be too long and serious injury could occur. And so this is why the reflexes work so quick this way. There is an inhibitory portion right here that actually stops the reflex from continuing over and over again. You can actually sit there and train yourself not to respond to a reflex. So the central nervous system can sit there like people walking on hot coals. If you psych yourself up to walk on a hot coal, you can actually do it and not actually have the reaction that most people do of not wanting to step on it or pulling your foot away or something like that. So you can't override a reflex. But if you're walking along and you step on a hot coal and you're not prepared for it, you can't stop the reflex. It's going to happen too fast. And then, by the time you think about it, it's too late. Your body's already reacted. So these things are actually about protection. Withdrawal reflex, this is a little bit more complex. So this is the classical one that I was talking about with pain. You step on a sharp tack or a Lego, and so you're going to have the sensory input that's going to go in. It's going to say, ow. And then what's going to happen, there's going to be a signal sent to the brain at the same time that there is an efferent impulse out to skeletal muscle to tell the leg to withdraw from what you just stepped on. And it's going to actually involve multiple different areas of the spinal cord that are going to do multiple muscles that are going to sit there and actually be involved with withdrawing the leg from that sharp object you stepped on. So this right here is what's known as a withdrawal reflex. And one of the things that we can get into is these things can actually be sometimes ipsilateral too. So, one thing is you might step on something sharp and the reflex is for you to pull your leg up. At the same time, you might stretch your arm out looking for something to grab onto really quick to where you can take weight off of whatever sharp that you're stepping on. They don't have one to show ipsilateral, so that's why I kind of threw it in there. But the other one is cross extension. So stepping once again on that sharp tack or on a Lego. This one right here, you have your normal reflex that we just showed here of the muscles withdrawing, but on the contralateral, you also have a reflex that goes to the other side that says, put the other leg down, because if one leg's in the air and the other one tries to come up in the air, you're going to fall. So this is a protective reflex that with cross extensor that makes you withdraw the leg that something sharp is on, like the Lego, and you hope there's not a Lego on the other side because your other foot is going to go down contralateral really quick. And you do it so quick that you don't realize what's going on. Your body's going to sit there and do this automatically. And then after the pain and everything, then it's going to register in your brain what's actually happened. Moderation, so the CNS, the brain, can actually moderate these. If you have an injury, say like we actually have a transection of the spinal cord between the thoracic and lumbar sections, it is possible in the hind limb, even though that the spinal cord's fully been transected, to still have reflexes, and this actually comes in a lot of our trauma cases. The thing is that the central nervous system of the brain actually moderates reflexes, and it says, okay, I sense that there was a reflex and you did it, now you can stop doing any more reflexes. And if you actually cut the brain off from the spinal cord, what happens is we get hyperreflexia. So this is actually where we end up having this reflex arc gets damaged, and so the reflex is going to happen again and again and again and again. There's nothing to actually stop it. If there's trauma where there's directly a reflex arc, then we have no reflexes whatsoever. So these become clinically important when we start looking at reflexes with neurological trauma. We can figure out, well, where exactly did that trauma occur at? Do I have hyperreflexes or do I have a lack of reflexes? And so that can tell me where it's at. And once again, the moderation of this is the central nervous system. They can override reflexes. Other, well, some very clinically significant reflexes, really quick here, is palpebral reflex. So this is when something's coming at or coming towards the medial canthus of the eye and touches it. We use this a lot in anesthesia to gauge depth of the patient. And so whenever you see a palpebral reflex, you're doing the medial canthus, you tap on it or you get close to it, and the eye blinks. That's the way that it's supposed to work. We have what we call the pupillary light reflex. This is where we shine a light in the eye. Normally, if you shine a light in the eye, one pupil is going to constrict. And then we have the contralateral, the opposite pupil constricts. So they're both supposed to constrict together. We also have things such as the meninges reflex. This is where you take your hand and you move it towards somebody's eye and they blink. There's also things like gag reflexes that are very important, because a gag reflex is important when we go to intubate a patient for anesthesia. If they have a gag reflex, they're not ready to be intubated. If you force intubation, they can actually vomit. Swallow reflex is also very important, because when they're recovering from anesthesia, we don't extubate them or remove the endotracheal tube until we're sure they can actually swallow.